Good morning, everyone, and good afternoon. Welcome to Indoor Ag Science Café. This is Chieri Kubota. I'm a professor at the Ohio State University. This is an extension outreach program of OptimIA program, or OptimIA project, funded by USDA Specialty Crop Research Initiative. So this is a space to discuss technologies and then also scientific findings or learning something basic if you are new to this space. So I really welcome everyone today joining us Indoor Ag Science Cafe. So as usual, I want to introduce the upcoming schedule of cafe series. We do Tuesdays, most of the time, Tuesdays, 11 a.m. Eastern. And then today I'm going to be the speaker talking about basics of lettuce tippan. And then March, we have Neil Matson from Cornell University talking about interesting tool or useful tool, and then also his scientific thought about the light quality effect, specific light quality effect. And then we will have April, Simone Valdesona Souza is going to talk about the consumer survey result. And then May, we have another basic information, but engineering basics of the CFD provided by Dr. Murakachira, University of Arizona. And then we will have still a couple of space for the June 2nd technology showcase. So I like you to consider if you are interested in participating in that June cafe. So with that, I'd like to move on to today's topic, lettuce tippan basics. Again, I'm a professor at the Ohio State University. I'm also the director of the new center called Ohio Controlled Environment Agriculture Center. And then I'm so glad I have this opportunity of talking about tippan, which we are working on over the past couple of years, or maybe more, including other crop. So tippan is a long-term issue. It's interesting, the research, that many years ago research, or a report suggesting that issue. And then there are so many milestone research projects done, and then we are appreciating that because some of them are foundational research to become a practical solution for tippan. But today I want to emphasize that it is still ongoing problem, and growers still, don't know why this is happening, or do not have practical way to avoid that risk. So so that's basically the situation right now. So let's start over the talk. So today's topic, I like to focus 4 things. One is the goal is you understand that tippan is a localized calcium deficiency. It's not because calcium is not available in the root zone, but because it's not delivered to the needed area. And then also mitigation measures, I'd like to introduce what kind of measures available, and risk assessment approach we are working right now. And I'd like to know if you are interested in collaborating with us to evaluate your environment. So anyway, try to be finished probably 15 till so that we have a lot more time to discuss. Anyway, let's go. So the tippan typical symptoms, many of you probably already familiar with, so just just a reminder, I guess. So it's a browning of edges of tips of leaves, just like that, just like this. And then it's always starting from growing tip. That's the unique thing of tippan. There's a reason for that. And whenever plants are growing very fast, and then that's the time you start seeing tippan. And then because plants start growing really fast to enter the exponential growing stage, therefore you tend to see that toward the end of the production time. US growers or North American growers typically grow 6 weeks or so to get the full head lettuce in the optimum condition. So in that situation, you get to see tip-pan. You might see tip-pan during the 5th or 6th week after seeding. So it's really close to the harvest. So that's the problem, you know, looking good, and suddenly tip-pan. So as I said, it's starting in growing shoot tip. And then the typical symptom is a very much deformed leaves as they unfold or expand, because it started in young stage, so that cell wall cannot expand as normal. So therefore it just deformed. We know that it's a calcium deficiency. So Neil Matson, Cornell University did an excellent extension article in eGrow 2015, talking about the difference between tip-pan and edge band. I call edge band, but I think he called outer leaf marginal necrosis. Some people call tip-pan also, so that's why the confusion coming, you know, that when you are talking about tip-pan, you need to make sure you are talking about the real tip-pan coming from calcium deficiency, localized calcium deficiency, or talking about the salt stress induced edge band, because two things are different in terms of symptom and then also causal factor, just like as I said. So the edge band is more like, you know, fully unfolded leaves, and then edge gets the necrosis, necrotic tissue, but tip band starts from beginning, therefore it gets really deformed a shape. So that's, I think, easy to identify. And edge band doesn't, may not happen in young leaves, but happen old leaves too. So that's why it doesn't get deformed. Oops, did I skip? No. So the calcium function, and then deficiencies in plants, so the basic part. So it is a very important constituent of the cell wall. So when calcium is not available, cell wall doesn't develop. So that's why it get collapsed or ruptured, and then having the issue of, you know, burning the cells and tissue. And in addition to tip-pan, calcium deficiency is known for other organs or other species. For example, tomato has the blossom end rot, which is also localized calcium deficiency. Calcium is very important, involved in many, you know, enzymatic activities or membrane function. So when it is not enough, then you have a huge issue. And then we know that in hydroponics, we usually have good amount of calcium, adequate or optimum amount of calcium in the nutrient solution. So it's known as more environmental nutrient disorder, not actual nutrient lacking situation. So the typical mistake I have seen among growers who don't know what is causing calcium deficiency in the growing shoot tip is that growers tend to put higher concentration of calcium in a nutrient solution, and then causing different issues such as potassium or magnesium deficiency, because those are all cations, and cations tend to compete each other to get, you know, absorbed by the plants. We call a cation antagonism, and then that is a well-known, how do I say, well-known fact in plant nutrition. So, so a little bit more about calcium. And calcium is again a cation, therefore it competes with magnesium and potassium. And then also calcium uptake is reduced when pH is low. Why? Because low pH meaning more hydrogen ions. Hydrogen ions are also cation, therefore it competes with calcium. It's very much mass flow driven uptake. So mass flow meaning a massive amount of water flow. In plant system during the day, it is really transpiration is a mass flow. During the nighttime, when transpiration is minimized or suppressed, root pressure becomes the driving force of the mass flow in the xylem. And then xylem is the pathway for the calcium to get transported, not the phloem. So that means calcium, when it is delivered to existing leaves through the xylem flux driven by transpiration, there's no mechanism to get calcium available and unload and then deliver to somewhere needed more. So that's why we call calcium is not mobile. And that's why the deficiency usually starts in growing young leaves rather than old leaves. So unlike, if you are talking about nitrogen deficiency, you know that deficiency starts in old leaves because plants can take the nitrogen from the old leaves and then provide that needed nutrient to the young, growing young leaves. But calcium, you can't do that because it's a xylem base, not phloem transported. So always start in young leaves. So, so that it's not really just availability issue. It's more like a demand and availability or supply issue. So that's the unique thing of TIPAN. And then high risk of TIPAN, unbalanced supply and demand of calcium. So that whenever calcium demand, so the needed amount of calcium to support the growth is exceeding by what is available or what is coming by xylem mass flow, then it is the high risk of TIPAN. So as you know, the transpiration and then root pressure is the driving forces for the calcium supply to the growing, young leaves. And then calcium demand is coming from growth, right? As I said. And then for those, functions, there are so many different environmental factors. So that's why understanding TIPAN is very, very complex, because you think the growth rate is reduced, but if the supply side transpiration is not enough, then you still see the TIPAN, or your condition is driving good transpiration. But if the growth rate is more than what the supply can meet, the demand side, then you get TIPAN. So it's really, you need to understand both to understand TIPAN and manage TIPAN. All right. So a little bit more about transpiration, because obviously transpiration is an important thing to understand TIPAN. So it's an evaporation from plant tissue. It's an evaporation of water from plant tissue. And then plants are not pumping out water. It's just open stomates widely and then let the water evaporate from stomatal cavity. So that's what plants are doing. Because it's evaporation, it is affected by many environmental factors. As you know, wind is a very strong factor promoting evaporation. Humidity or VPD, vapor pressure deficit, air temperature, leaf temperature, or radiation, solar radiation, thermal radiation, both affects the evaporation rate. And then plants have a regulation by themselves, closing, opening stomates. But under the optimum conditions, you don't want to let plants close stomates because that would reduce another important gas exchange, which is photosynthesis. So ideally, plants keep the stomates open, let the water evaporate, and then let the CO2 come in for photosynthesis. So that's the optimum condition. And then the last thing, we don't talk too much, but I think it's very important to understand, particularly for indoor setting or the greenhouses who use a lot of supplemental lighting. So electrical lighting spectrum is usually probably up to 800 nanometers. Most of the cases, 700 nanometers. It's very different from sunlight. Sunlight is all the way to probably close to 3000 nanometers. So the sunlight has more energy to drive transpiration rate, evaporation of water, compared to electric lighting, even at the same PPFD or DLI, daily light integral. So that's very important, to understand indoor or supplemental lighting conditions compared to just natural sunlight situation. So more transpiration under sunlight, and then less transpiration in the electric lighting when the light intensity is the same. All right, so how do you avoid tip bands? You're probably doing this, and then in that case, you don't see this as much of a problem, but I'd like to go over for those who need this information. So easiest one, avoiding something very sensitive. If you look at lettuce varieties or cultivars, you see huge range of tip band sensitivity. So selecting something relatively tolerant is a good idea. However, sometimes, you want to grow this cultivar, or a customer want to grow this cultivar, but that might be not a good choice in terms of tip band. So in that situation, you have to do some sort of mitigation procedures. Number one, slowing down the plant growth. This is the historically traditional approach, lowering temperature to slow down if you can do that, or limiting light intensity, shade cloth, or reducing supplemental lighting hours or intensity if you can, so that you can slow down the growth, reduce the demand, so that meeting the supply side of calcium. Harvesting early is a unique way, but actually it's very common in North America. And then instead of waiting to the mature full head lettuce, they harvest in a mini size so that they can avoid tip band risk. Enhancing mass flow, enhancing transpiration has been practiced in greenhouse, but not so much in indoor ag setting. And then I talk about the data. And then foliar calcium spray is also suggested in the past. And then some growers, I think, use that. All right, so the limiting light. Great work was done in Cornell University. Lou Albright, AJ Both back then in Cornell did a lot of studies for tip band management, and then suggested, here is the threshold you should avoid beyond that for this type of cultivars or that type of cultivars. It's very useful. So for example, romaine lettuce, I believe 12 moles per square meter per day is a sort of suggested threshold. And then other types of lettuce not exceeding 14 or 16 is a good idea. Because greater DLI pretty much faster growth. So that's why greater DLI would increase the risk of tip band, unless there is effective mitigation is in place. However, I like to mention that because this is a group of indoor vertical farm growers and technologists, that those thresholds may not be applicable to indoor because of the conditions, because of the light quality, because of the light we use does not have a thermal energy as much as sunlight. So we should look at the thresholds specific to indoor setting one more time. But again, this concept of reducing or limiting light is very, very helpful. And then thanks to those who worked in early, I guess, 90s for this study. And then this is actually some of the outcomes of the research done in Cornell. And then original paper is AJ Both, 1970, sorry, not that old, 1997. I'm just checking if AJ is here, but It's a great work. And then the particular chart was copied from more recent publication put together by Chris Curry at Iowa State University, showing the growth rate of lettuce over, I think, 6 weeks of time under different DLIs. So starting at 8 moles per square meter per day all the way to 22. And then if you look at the tip burn incidence, 20 or 22 moles per square meter per day DLI, they got tip burn. So you can see the clear threshold somewhere between 16 and 20 for this particular butterhead lettuce. So another way, right, to reduce the growth so that calcium demand is reduced is slowing down by temperature. And then this is an example temperature response curve of crop. And you can see that peak temperature in this case, 25. So this is crop specific or cultivar specific. So don't take this. This is a universal value. But that is most likely around this temperature is the sort of minimum temperature. So that the typical, mountain shape or shifted mountain shape can be created knowing base temperature and then also optimum temperature. And then once you know those, important temperatures, then you can create linear range starting from the base temperature 5 degrees C in this example, ending 25 degrees C in this example. And then that would tell you, every single degree C reduction, you can achieve roughly 5% growth reduction. So just slowing down. So that means you need to expand the production time to meet the same size. But because that moment by moment growth rate is reduced, so that you can expect the lower risk of tip burn by doing this. So this one is a sort of, again, one example of growth curve. As you know, plants grow exponentially, right? So they initially very, very slow, and then maybe several weeks after they start getting into very fast growing phase. And then that huge, increase of growth rate actually gives a big risk for the tip burn. Again, calcium deficiency or tip burn is coming from the balance between growth and then also transpiration or the supply of the calcium. So typically this fast growing phase is the highest risk. So you want to avoid that. So that's the practice I see in many, many North American indoor farms and sometimes greenhouses doing. So the harvesting early, so that avoiding tip burn. But harvesting early meaning the crop, your plant size is very, head size is very, very small because it's before getting into the fast growing phase. So what they do is usually having a much higher planting density, so the high density production, so that per area based productivity is the same, and in short cycle, so that annual yield is not going to change much. So that's the strategy. However, a group of OptimIA project members actually looking at this practice really closely in terms of growth rate, in terms of operational costs, material costs, and then utility costs to see if this practice actually making less profit for the production. So the members are Muraka Chira, who is the engineer, and then his student, Casey, and then myself, and then the economist, Simone Barre Souza, in Michigan State, working together to look at this. And then I hope we have opportunity to share the finding. It's really, really cool study, meaningful study, I would say. All right, so how do we grow as avoid tip burn, right? So we talked about slowing down, and then we talked about harvesting early. So I like to talk about enhancing mass flow, even though this is not easy to introduce in indoor farm situation, but it's a quite established approach in greenhouse, because greenhouse has much bigger headspace. Therefore, putting something over the headspace is a more feasible in a greenhouse situation. So when you enter greenhouse like that, you can instantly tell this is for leafy green without looking at the crop. Usually you look at the crop first, but if you ever do that, you probably can tell this is for lettuce or some kind of leafy greens having a tip pan issue. Important thing is it is the downward vertical airflow. Downward, not upward. There's a vertical airflow fan marketed using that name, vertical airflow fan, but going upward. So that doesn't work for tip pan. You have to go downward, hitting that crop. So the air goes directly over the crop. So that's why growers use like this kind of seeding fan, like your warehouse might use, or putting the regular HAF fans, which is designed to go sideways down. So that's for tip pan management. So why you can get tip pan mitigated with this downward airflow? Because over the plants, there is a very much stagnant zone of air, which is limiting gas exchange. We call boundary layer. Again, it's because of the friction between the air and then also the surface. The air doesn't move easily as, much higher headspace. So that boundary layer limit the transpiration. If you have airflow, that boundary layer gets, thinner and thinner, and then promote gas exchange, transpiration, more transpiration happening from the leaves. And then we know that optimum air current speed is about 0.5 meter per second, which is about 1 mile per hour. So it's a gentle air. It's not really fast speed. It's just enough to get leaves moving like this, gentle. So that's really good airflow. And then Yoshiaki Kitaya, professor in Osaka Prefecture University, my mentor in the past also, he did a lot of work in air current speed and gas exchange of the plant. And then it shows that transpiration increase as increasing air current speed above the plant, and then boundary layer declines. So that's really, he actually measured those. And then this is my sort of simple presentation of boundary layer limiting the gas exchange or transpiration because it creates additional sizable resistance to that gas exchange. All right, I like to talk about this always because I really appreciate those two people, scientists / engineers, did that study back in 90s. Eiji Goto was, I think back then, assistant professor at the University of Tokyo. And I was a graduate student back then, not in Tokyo, but different university. But I was familiar with his work. What he did was quite unique. He had that tip pan issue, right? And then he wanted to see if targeted airflow around the shoot tip actually removed the tip pan or not. So he set up in a sort of artificial, environment. It's not greenhouse, and then put those, tubings, airflow targeted right onto the shoot tip, and then compare the tip pan incidence, growth rate, and all that. And then he successfully showed that tip pan incidence was reduced or almost eliminated by having that targeted airflow around the shoot tip. Airflow, reduced boundary layer, increasing transpiration around that, shoot tip. Lou Albright, who was a professor in controlled environment agriculture at Cornell University, engineering, did a study of the airflow in the greenhouse, and two together, I think, communicated apparently, but or collaborated, because I know Eiji Goto spent some time in Cornell while he was working in the tip pan. But anyhow, the great thing of Cornell contribution was practical solution of tip pan by just redirecting the airflow to downward in the greenhouse. So this is A.G. Goto's research showing that, the schematic diagram, airflow, targeted airflow around the shoot tip. And then you can see that without airflow, they get up to 100% tip ban. With airflow, almost no tip ban, which is quite remarkable results. And then also they tested calcium concentration of outer leaves and inner leaves. And then here, two bars showing calcium concentration of the lettuce plants without targeted airflow around the shoot tip. Therefore, inner leaves have very much low calcium concentration. Outer leaves have good calcium concentration because outer leaves, have widely open, right? And then exposed to the environment. Inner leaves are sort of enclosed or in a sort of dented area, I would say. Therefore, more boundary layer accumulation in the shoot tip area. Therefore, less transpiration, lower concentration of calcium. If you have airflow targeted around the shoot tip, look at this increase almost several times and then close to the level of outer leaves. So that is explaining why tip ban could be mitigated. He also tested nighttime only, airflow to see which phase of the day, daytime, nighttime, is more effective in terms of enhancement of the calcium uptake by airflow. So he showed that it still increased the calcium uptake by inner leaves, but the impact is much bigger during the daytime because obviously stomates are open during the daytime and closed during the nighttime. But during the nighttime, still non-stomatal transpiration. So that all the surface actually lose some water by evaporation. So that is the showing the difference between this and that, the increase by airflow. But impact is much higher during the daytime. All right, so vertical downward airflow is very important. And then greenhouse, you can do that. And then again, the airflow is hitting the shoot tip area is very important. And then the target is around 0.5. I think 0.3 is also good, but I would recommend 0.5 is good to get the maximum, right? And then too much airflow, of course, it's not good. So it's a good gentle airflow. All right. So Foliar calcium spray is something we know as recommendation from horticulturists, greenhouse growers, or greenhouse researchers. To be honest, it's not my favorite approach because I have to wet the plants and then I have to spray manually several times a week. I wish it could be automated, but I'm not sure if it is easy to do that. But despite that, always wetting the leaves is kind of concern for me. Although researchers say that the calcium chloride actually antifungal, therefore it doesn't cause any issue. But still, I guess, but it seems like working. So I like to show research reported very recently. So the calcium concentration after 400 parts per million, milligram per liter, significantly reduced tip ban. I think this is butterhead lettuce. However, it seems to reduce the growth rate also. So that might be another issue for this approach. But if you wish to do that, I think that is the concentration recommendation coming from that research. And then you have to do that 2 to 3 times per week application. So there are mitigation measures, And so what about indoor specific mitigation in addition to those I talked about? So one is humidity management. In fact, my group worked on that several years ago when we started working on different crop, but having tip-pan problem, which is strawberry, greenhouse strawberry. In our climate, always have the tip-pan, calyx pan, calcium deficiency. And then we started doing humidity management because downward vertical airflow really didn't work with that structure of canopy of the strawberry. So we started working on that. And then we done that effort was done many years back, probably 90s or 80s by Ted Tibbetts, which used to be called Mr. Tip-pan or Dr. Tip-pan, professor at University of Wisconsin. There are so many people contributed tip-pan study, but anyway, so the humidity management works, but you need to achieve low humidity during the day, super high humidity during the night, and that is a problem because it increases HVAC cost. One of the OptimIA group is HVAC engineer. You probably know who it is, but she always talk about humidity issue. And then whenever I say high humidity or low humidity, it's not easy. Again, high humidity side in the ambient temperature, like 18, 20, 22, it's just a huge issue of foliar disease. So it may not be a good solution for indoor ag people, although it works. We know it works, but I stopped recommending indoor growers because I know it's very challenging. But I still recommend strawberry growers because strawberries are grown relatively low night temperature, which is off from the optimum, for example, botrytis growth. So we could do that and then actually successfully or almost successfully continuing doing that approach. But anyway, so if you increase the humidity to the very much saturating condition during the night, it actually maximizes the impact of root pressure. So the xylem is going to be maximum pressure, sort of get swollen type of situation. So that squeezing calcium, pushing calcium containing xylem solution, xylem sap to the growing tip. So that works, that provides the needed calcium and then reduce the tip-pan risk. So that's the concept of increasing humidity at night. Reducing humidity during the daytime is generally enhancing transpiration rate. But anyway, it's difficult in the indoor situation. And then airflow design inside the growing system. This is the approach Murakachira in University of Arizona and his group is doing. So in the future, maybe when he talk about the basics of CFD, computational fluid dynamics, the simulation of airflow, in May, he might be able to include that information in his presentation. He actually did a quick presentation in the past in a cafe series a couple of years ago, if I remember correct. But anyway, so that's, you need to custom design, right? The shelving unit. So that may not be your solution. And then the interesting approach reported also by Eiji Goto, now in Chiba University, but back then in University of Tokyo, he was talking about shorter cycle. I think he did like a 3 hours, if I remember correct, day and night cycle instead of 24 hours to mitigate the turgor so that cell rupture can be mitigated, which is the sort of initial problem after calcium deficiency in growing tissue. But anyway, it's very interesting, but the particular paper shows the mitigation only by 3 to 4 days delay. So this may not be enough for some situation, but I think it's interesting to take a look if you are interested in doing this. And then the last one, I think we need a lot more information for this. And actually we are working on this approach also, like quality, how we can use that approach because it's a unique capacity indoor farming situation has. So hopefully I can give some update about this approach in the future. But I'd like to talk about risk assessment before I stop this presentation. So a few more minutes. So we started doing that because mitigation is important, but if there's no risk, why you have to do mitigation? So, but there's no way to assess the risk level for the tip pan. So we started working on that. Our basic approach is understanding transpiration and then understanding growth. And then we are currently doing a tool-based direct measurement of evaporation to understand potential transpiration. And then a reference model, sort of standard or standardized model-based growth assessment, because there is a huge range of the growth and cultivars. And so it's more sort of simplified growth model-based assessment of the growth rate. And then together trying to understand the balance of calcium supply and calcium demand. And then get some kind of tip pan risk index so that growers know about the environment, if it is high risk or low risk in terms of tip pan. So again, the risk assessment index, we want to, based on the direct measurement of evaporation. Why direct measurement? Because transpiration is so many factors, and then individual factors cannot tell what is the likely transpiration. So that's why we decided to do direct measurement. And then a whole bunch of measurements my former graduate student, Gio Papio, did, and then get that good idea of what is the range we have in that growing environment, typical, a growing environment indoor, and then what is the range we might want to call low transpiration or the evaporation, and then what is the high evaporation rate. So this is sort of our summary. Less than 10 moles of water per square meter per hour evaporation, which is quite low. Therefore, there might be high risk when growth is not, meeting that supply. Greater than 20 is good because it's quite high. And growth side, we use a sort of reference growth model. Thanks to my colleagues in OptimIA group provided data, as well as my graduate student. We now have a very simple model based on the light interception, how much light plants are receiving, and then what condition of temperature and CO2. From that, we can compute, without measuring actually, compute the sort of theoretical growth rate. And then together, we should be able to come up with tip pan index. And then I like to have index something like that. Greater than 1 is a high risk. Smaller than 1 is a low or moderate risk. So that way, I think it can help to decide, selecting certain cultivar or, introducing some mitigation measures in your facility. So just quickly, this is a condition we selected for doing a cultivar trials. My graduate student did that, John Arrow, graduate student. And then just showing the actual, evaporation rate, which is very low, and then growth potential from the standardized model, very high. So two together, it's a huge high risk of tip pan. And then you can actually tell when likely high risk starts. And then this assessment tells me, because of the growth model, starting around day 28 to day 30. So that from the seeding date. So that's quite useful. And then he could show that, the range of tip pan. We selected relatively sensitive cultivars in addition to some of the reference cultivars like Roshai, which is very tolerant to tip pan. But we could show that all plants, having tip pan, because it's a high risk tip pan environment we selected. So it was very successful. And then now we are working on mitigation. And then I'm hoping to share that, or graduate student John Otto can share that in the future cafe meeting. So if this is a problem, I like to help, and we like to help. If it is not your problem, that's fine. Enjoy your production, it's great. But if this is a problem, what we can do is we can assess your level of tip-pan risk. So that's one step we can do. And the great thing of this direct measurement of evaporation is that I don't need to know your conditions. I can just send you the tool, and then you can just tell me the number. And then I can tell it is high or low, right? Growth analysis can be done also the same way. I can just send, how to plug in the numbers, and then you can just come up with a number, and then two together, you can tell if it is risky environment or non-risky environment. And then if it is risky, we can talk about mitigation. And then maybe a new design of the rack for the future facility, or it could be something more standard, like lowering temperature or lowering CO2 or things like that, or harvesting early, things like that. So if you are interested, please let me know, and then I'm happy to work with you. Another thing I like to inject here, if you have any capacity of simple sensor development, I really like to work with you, because I want to. So what we are using for the evaporation measurement is a direct measurement and then very high-tech Petri dish measurement. So instead of using that, actually low-tech, I like to have some kind of sensor capacity, assessing the dynamic change of the evaporation rate. So if you have that capacity, I like to work with you. So I guess that's all. Thank you so much. First of all, my lab members, John Arrow and Gio Papio, particularly, who did the work related with tip-pan, and OptimIA members, Eric Ranco, Roberto Lopez, Mura Kachira, who provided lettuce growth data for my model, and then Simone, and then Mura, and then myself working together almost every other week for working on that modeling and the assessment of the profitability. So, and then USDA, and then other companies, all the companies who provided in-kind support. So with that, I'd like to thank again everyone, and then stop recording here.